Block 1, Unit 3, Macrophage Biology and Salmonella Infection. Salmonella-related diseases have a huge impact worldwide, both in first and third world countries. Despite improvements in hygiene and availability of antibiotic drugs and vaccines, every year around 100 million people worldwide are likely to suffer from food poisoning or typhoid fever. Understanding how Salmonella is able to bypass our immune system and cause infection is a key global health need. Suffering from food poisoning is an unfortunate experience that most of us are likely to encounter during our lifetimes. One of the most common causes of food poisoning is bacterial infection from Salmonella, typically by ingestion of contaminated food or water. After passing through the digestive system, Salmonella are recognized by macrophages, the sentinel cells of the immune system. These cells are highly specialized to identify and remove harmful substances, cellular debris, and bacteria. Macrophages surround and engulf Salmonella. The macrophage then contains the bacteria within an intracellular compartment called the Salmonella-containing vacuole, SCV, and lowers pH levels that normally effectively becomes an acid bath designed to dissolve and kill the bacteria. However, Salmonella have developed ways to evade this system and survive within the macrophage. The innate and acquired immune systems. The human immune system is composed of multiple organs that protect the system from foreign substances or cells. The immune system is typically divided into two categories, innate and adaptive, although these distinctions are not strictly mutually exclusive. Innate immunity refers to nonspecific defense mechanisms that come into play immediately or within hours of an antigen's appearance in the body. An antigen can be any foreign substance, including toxins, bacteria, and viruses. These defense mechanisms include physical barriers such as skin, chemicals in the blood, and immune system cells that attack foreign cells in the body. The innate immune system response is activated by the chemical or molecular properties of the antigen. Adaptive immunity refers to antigen-specific immune response. In other words, this component of the immune system mounts a response that's appropriate for the specific antigen present in the system. The adaptive immune system is more complex than the innate. The antigen first must be processed and recognized by the adaptive immune system. Once an antigen has been recognized, the adaptive immune system creates an army of immune cells specifically designed to attack that antigen through the production of antibodies. Adaptive immunity also includes a memory that makes future responses against a specific antigen more efficient. This memory is what makes vaccines effective after the first exposure to the antigen, like inactivated measles virus. Macrophages are specialized cells in the innate immune system that are involved in the detection, phagocytosis, and destruction of bacteria and other harmful organisms. Phagocytosis is the process by which macrophages take up foreign substances like bacteria into compartments called phagosomes. This is a receptor-mediated process and involves interaction between the pathogen and the actin cytoskeleton of the cell. The phagosome then fuses with a lysosome, thereby creating a phagolysosome. This fusion produces a more acidic pH that activates enzymes in the phagolysosome and assists with the degradation of the pathogen. Waste is then expelled from the cell for other cells in the innate immune system to further degrade. Alternatively, antigens from the pathogen are recycled back to the surface of the macrophage for recognition by the acquired immune system. Macrophages originate from blood monocytes that leave the circulation to differentiate in different tissues. There is substantial heterogeneity among each macrophage population, which most probably reflects the required level of specialization within the environment of any given tissue. This heterogeneity is reflected in their morphology, the type of pathogens they can recognize, as well as the levels of inflammatory cytokines they produce. In addition, macrophages produce reactive oxygen species, such as nitric oxide, that can kill phagocytosed bacteria. The heterogeneous nature of these cells may not solely be the result of their differentiation process, but it is likely to be inherited from their monocyte precursors. Macrophages migrate to and circulate within almost every tissue in the body, patrolling for pathogens or eliminating dead cells. They are able to enter tissues from the bloodstream through a process called extravasation. Macrophages are able to detect products of bacteria and other microorganisms using a system of recognition receptors, such as toll-like receptors, TLRs. These receptors can bind specifically to different pathogen components like sugars, LPS, RNA, DNA, or extracellular proteins, for example, the protein flagellin from bacterial flagella. And upon binding, the macrophage becomes activated. Upon activation, the process of extravasation is started, whereby a macrophage begins to adhere to the endothelial cells of the vasculature and then migrates into the tissue, either between endothelial cells, paracellular, or through the cells themselves, transcellular. Some bacteria exploit the immune system by entering the host cells and residing there. Advantages of entering a human cell include providing the bacterium with a ready supply of nutrients, protecting the bacteria from antibodies and other body defense molecules. In addition, some pathogenic bacteria invade phagocytic cells, neutralize their killing ability, and turn them into a safe haven for bacterial replication. Or kill phagocytic dendritic cells once they are engulfed and prevent those dendritic cells from activating other cells required for adaptive immunity. Salmonella, Shigella, and some strains of E. coli, for example, enter epithelial cells of the colon. These bacteria, like many involved in infection, have the ability to co-opt the functions of the host cells for the bacterium's own benefit. This is done by way of bacterial secretion systems that enable the bacterium to directly inject bacterial effector molecules into the cytoplasm of the host cell in order to alter its cellular machinery or cellular communication. The most common type is the type 3 secretion system, T3SS. A secretion apparatus in the cytoplasmic membrane and cell wall of the bacterium polymerizes a hollow needle that's lowered into the cytoplasmic membrane of the host cell, and a protein is then delivered to anchor the needle to the host cell. Effector proteins in the bacterium can now be injected into the cytoplasm of the host cell. The delivery system is sometimes called an injectosome. Salmonella use a T3SS to prevent fusion of the phagosomes with lysosomes within the host cell. When these bacteria contact the epithelial cells of the colon, the type 3 secretion system delivers proteins into the epithelial cells, enabling them to polymerize and depolymerize actin filaments. This cytoskeletal rearrangement is a key part of the pseudopod formation in phagocytic cells and is what enables phagocytes to engulf bacteria and place them in a vacuole. Thus, the bacterium, with its invasins, is able to trick the epithelial cells into behaving like a phagocyte and engulfing the bacterium. The bacteria then replicate within the host cell. Salmonella use a type 3 secretion system to inject intestinal epithelial cells with effector proteins that stimulate actin rearrangement and cause the epithelial cell's cytoplasm to ruffle up and engulf the bacteria. The Salmonella pass through the epithelium where they are engulfed by phagocytic macrophages. Once in the phagosome of the macrophage, the bacterium uses its type 3 secretion system to inject proteins that prevent the lysosomes from fusing with the phagosomes, thus providing a safe haven for Salmonella replication within the phagosome and protecting the bacteria from antibodies and other defense elements. By injecting flagellin into the cytoplasm of the macrophage, the Salmonella can also eventually kill the macrophage by inducing apoptosis, a programmed cell death suicide. Molecules injected into the intestinal epithelium also stimulate diarrhea. Advantages of inducing diarrhea include flushing out normal bacterial flora so there's less competition for nutrients, and better enabling Salmonella that are not attached to host cells to be transmitted to new hosts via the fecal-oral route. Salmonella has evolved over the years to survive and even reproduce in the SCV. Although the existing viewpoint is that the Salmonella somehow manages to counteract the acidic surroundings and maintain neutral conditions, it is still unclear how Salmonella is able to survive after being engulfed by macrophage. Dr. Smarajit Chakraborty and Hideaki Msusaki from the lab of Professor Linda Keeney discovered that Salmonella does not neutralize the acidic environment, but instead it acidifies itself to match its surroundings. The learning goals for this unit, macrophage biology and Salmonella infection, are: distinguish between the innate and acquired immune systems in terms of stimuli and cells. List the steps of phagocytosis. List the steps involved in Salmonella infection of macrophages. Describe the mechanism by which Salmonella is protected from acidification by the lysosome.